So our next speaker is a recorded session. Unfortunately, our speaker couldn't attend today. Presentation from Sergey Rakhilin from C-Path, one of C-Path's more recent consortium in rare disease, the critical path for sickle cell disease. Sergey works to push that consortium and to improve and streamline the regulatory approval process for new treatments for sickle cell disease. Sergey comes from industry with previous work at various pharma company, and he worked a lot in drug development. So please virtually welcome Sergey.
Good morning, ladies and gentlemen. My name is Sergey Rakhilin. I'm an executive director of Sickle Cell Disease Consortium at C-Path Ltd. I would like to thank the organizers of this workshop for inviting me to present. Our consortium is relatively young. We're just 2 years old, and we would like to learn from the experience of other more advanced programs, including rare diseases, on the developing mutually beneficial collaboration with RDCA-DAP. Sickle cell disease is a very unique illness because we all remember it from the biochemistry book, and we think that we understand it quite well. It is a hereditary disease where a single mutation disrupts hemoglobin function. Approximately 100,000 of Americans suffer from that devastating disease every day. 4 drugs delivered to the market within the past 10 years. Treating one or another manifestation of the ailment. 33 companies are developing novel treatments of the disease. And importantly, almost half of these companies use gene modification approaches such as CRISPR, meaning that if we succeed in that approach with sickle cell disease, similar solutions will become available for other hereditary diseases as well. However, if you choose to divide total number of investigational clinical trials derived from clinicaltrial.gov site, run by industry within the past 10 years, by the number of drugs approved within the same period of time, and designate that ratio as a drug failure rate, you could notice that the ratio for sickle cell disease is approximately 2 to 3 fold worse compared to any other monogenic diseases, as shown on the right side of the slide. You can see that if you compare sickle cell disease failure in the phase 2 clinical trials only to similar failure rate in hemophilia, cystic fibrosis, Huntington, Duchenne, lysosomal diseases such as Gaucher and Fabry, the difference is quite striking. What are the factors that contribute or may contribute to the clinical trial failure most often? I listed several of those factors in the next slide as actionable items. Accelerate data sharing, provide endpoint harmonization of clinical endpoints, advance identification of biomarkers, and develop tools for patient stratification. These bottlenecks or actionable points correlate well with the goals of the Sickle Cell Disease Consortium that was organized by Critical Path Institute 2 years ago. This is a growing nonprofit organization consisting of 7 leading pharmaceutical companies such as Global Blood Therapeutics, Graphite Bio, Fulcrum Therapeutics, Novartis, Pfizer, and Takeda. The consortium provides neutral pre-competitive environment to address all of these questions to the FDA in the form of monthly meetings and face-to-face conferences. For example, one of the recent discussions led by Pfizer raised the question of generating a composite endpoint reflecting frequency of vaso-occlusive crisis, as reflected in the paper published by Deborah Pittman, evaluating longitudinal pain study in sickle cell disease eclipses trial. The endpoint relies upon data derived from electronic diary, patient activity profile monitored by actography, and variety of biochemical biomarkers such as cell adhesion, chemistry, cell counts, and inflammation. It was concluded that composite endpoint may improve identification and assessment of the vaso-occlusive crisis as a key measurement of the clinical efficacy as defined by Pfizer. Importantly, Sickle Cell Disease Consortium at C-Path Institute is uniquely positioned to collect and analyze the data for composite biomarker development. Another aspect of drug development where the consortium could be especially effective, as acknowledged by pharma and FDA alike, is patient certification. Sickle cell disease is known for genetic polymorphism in hemoglobin molecule, causing a single mutation forcing transformation of the 6th amino acid from glutamic acid into valine. However, this is not the only substitution that could cause a disease, as shown on the left part of the slide, where I listed beta thalassemia, hemoglobin C, and on the right side, I listed them all, including rare forms of the sickle cell disease, that is hemoglobin D, E, O, and Lipor. In reality, sickle cell disease could be seen as an umbrella for several diseases having similar phenotypic and genotypic manifestations. Hemoglobin gene analysis could be one of the approaches for patient stratification. Phenotypic variability could be considered as another approach for patient stratification, as demonstrated on this slide. Greg Cato, in his Nature review, identified at least 4 overlapping phenotypes indicated in this slide. The first one is called vaso-occlusive and is characterized by an increase in hematocrit, blood viscosity, and increased acute chest pain. The second is called hemolysis and vasculopathy. Is associated with elevated levels of bilirubin, LDH, pulmonary hypertension, gallstones, ulceration, and progressive kidney failure. The third is designated as pain sensitive and correlates with the overexpression of GDP cyclohydrolase 1 or GCH1, associated with high dopamine and serotonin uptake, and therefore making it very difficult to distinguish chronic from acute pain crisis. The fourth corresponds to the relatively high hemoglobin F expression, and patients bearing that phenotype are frequently asymptomatic. One of the key questions raised by industry is the concept of non-responders. For example, in this slide, we illustrate a hypothetical overlap of hydroxyurea, which is a fairly old drug developed more than 20 years ago, with all 4 phenotypic disease subtypes. Database analysis run by Sickle Cell Consortium could identify patient subgroups where drug was not effective and presents a clear unmet medical need. Voxelotor from Global Blood Therapeutics, which is a much more recent drug, or crizanlizumab from Novartis could be targeted in the very same fashion with a more relevant results, identifying even a more important unmet medical need, identifying patients who are not responsive to those drugs. In summary, Critical Path Consortium of Sickle Cell Disease and RDCA-DAP are actively collaborating to accelerate drug development of the disease, leveraging the learnings from rare disease activities in C-Path and other consortia such as Parkinson's, Duchenne, Alzheimer's disease consortia paved the path of data acquisition and analysis shown on that slide, using making it easier to use RDCA-DAP infrastructure for sickle cell disease. And our consortium intends to follow their footsteps to increase clinical trial success to cure the disease. Thank you very much for your attention.